Hello, everyone. My name is Caio Seguin. I'm a postdoc at the University of Melbourne and the University of Sydney. And this is my 2021 OHBM Symposium on Communication in Brain Networks talk. And today I'm going to give an introduction to brain network communication models and their applications. So network communication acts as a bridge between brain structure and function. And I like to illustrate this using this figure from a recent review by Soares and colleagues. So here they show a structural connectivity matrix mapped from white matter tractography and diffusion MRI, and a functional connectivity matrix mapped using fMRI bold signals. And what they show is that only a very small proportion of the functional connections in this matrix are actually underlined by structural connections. So if this is true, functional connectivity between structurally unconnected regions must be facilitated by polysynaptic paths in the structure. So here, this is a network representation of the connectome. And I've highlighted two regions in orange. And these regions are not anatomically connected, but we can imagine that they have a strong functional connectivity. And in this case, the question becomes, what are the paths, what are the sets of intermediate regions that facilitate communication between them? Here I'm highlighting two potential paths, but it's easy to see that these are only two out of many, many possibilities. So what are the propagation strategies that facilitate communication in the connectome? This is the question that brain network communication models tries to understand. So there are two main families of brain network communication models. The first one is called routing protocols, which proposes that signals going from a source region to a destination region do so by single communication paths. These are typically deterministic paths. In contrast, diffusion processes propose that starting from a source region, signals diffuse or are broadcast along multiple fronts in the network in more of a stochastic nature. So apart from these two families, diffusion processes on one side and routing protocols on the other side, there is also a family of models that I like to call parametric models. And these are hybrid strategies. They all have a parameter that controls their behavior, and the user can set this parameter. And by doing so, we can approximate either a diffusive behavior or a routing behavior or somewhere in between them. So these three families propose broad conceptualizations of neurosignaling. Each of them is populated by more concrete brain network communication models. So a model, as I'm defining here, is an algorithm to propagate signals between nodes or to identify communication paths between nodes. Here I want to quickly draw attention to shortest paths, which is the model that proposes that between two nodes, signals will be propagated via the most efficient path in the network. And this is by far the most popular model in connectomics. And it is often used without explicitly considering what it means to say that signals are exclusively being propagated via optimal routes. We can see that a lot of different other models have been proposed, and shortest paths is just a small percentage of these. These models fall along a spectrum from decentralized communication, on this side, from decentralized communication to centralized communication. So shortest paths is the quintessential centralized model. So what does that mean? Sending information via the shortest path is very efficient because by definition, it is optimally efficient. And that is very good from a metabolic point of view. So this would minimize the number of synaptic crossings, and it would minimize the energy required to propel signals along axonal projections as well. So you can see how shortest paths became a interesting model to use to understand neurocommunication. But recently, people have started wondering if this model is biologically realistic, because in order to know what the shortest path is, if you're a source region and you want to reach a target region, you need to know the entire network in which you are embedded. So this is what is called a centralized knowledge assumption. And it's hard to see how a system such as the brain, a decentralized biological system, would have elements with access to knowledge of the entire network topology. On the other hand, here we have decentralized models on the other side of the spectrum. And the quintessential decentralized model is just a random walk. So in a random walk, we propagate signals randomly. It's essentially running a Markov process on top of the network. Intuitively, this will be very inefficient. If you want to leave from a particular source region and reach a particular destination region, that is going to take forever. It's going to be inefficient, and it's going to be metabolically expensive because you're going to have to retransmit the same signal many, many times, which is also very error prone. On the other hand, it's a very simple and plausible model in terms of its implementation in a system where individual elements don't have knowledge of the whole architecture of the network. So there's this spectrum from decentralized to centralized. And nowhere along the spectrum, we seem to find a model that really hits all the right notes. So because of this, we still don't know which of these conceptualizations of neural signaling best describes what's really going on in the brain. And once you have a model which allows you to identify paths between nodes, you can quantify properties of communication under this model using network communication measures. So each model is associated with many, many communication measures. Well, sometimes just one-to-one association, but other times you can have many measures to one model. And I'm not going to get into the detail of what these measures are, but again, it's just to show that while many measures have been proposed in the literature, most of the studies still focus only on using shortest path routing. So now I want to showcase a few applications of network communication models. And the first one is a classic paper by Gogni in PNAS 2014. In this paper, he computed a communication matrix from a structural network. One of the key measures that he used was called search information, which is a measure based on random walks. And he saw that this analytical measure computed on the structural network could predict really well functional connectivity derived from fMRI. And what was really interesting is that search information is actually a measure that says that signaling doesn't flow exclusively via shortest paths. So this was really the first empirical evidence that perhaps communication is not really taking place exclusively along optimal routes. So this is a work from our group where we try to explore the properties of communication models being asymmetric. So what this really means is that sending a signal from node A to node B is not really the same as sending a signal from node B to node A. Because certain models A are decentralized, determining the path from one region to the other depends on the vicinity around nodes. Therefore, the direction of flow from A to B will identify a path, but from B to A, that will be a different path. So this is what we try to explore. So we start from a structural network, which is undirected because diffusion MRI cannot resolve axonal fiber directionality. And we compute a matrix of navigation efficiency. So that is the navigation communication model. And this matrix is asymmetric. So now we derive a measure of outgoing efficiency. So that means how efficiently every region can communicate with the rest of the brain, can send signals to the rest of the brain. and incoming efficiency, which measures how efficiently every region can receive signals from the rest of the brain. And we see that there were marked differences between incoming and outgoing efficiency. So here the senders are regions that are statistically better at sending information than receiving information, and the receivers are statistically better at receiving information. So one really cool thing is that the senders were predominantly sensory regions. So A1, S1, M1, and V1 were all senders. And the receivers were higher order regions, such as the precuneus or frontal regions. We compare this to the Margulies unimodal to multimodal gradient. And we saw that indeed unimodal regions were biased towards sending and multimodal regions were biased towards receiving. So the story here that we think is that just because of the embedding of the network, certain regions are embedded in a way that facilitates their quickly flushing out information that they receive from sensory organs to the rest of the brain, whereas other regions are better positioned to act as sinks and quickly receive that information and integrate multiple streams of information to facilitate high order processing. Then we try to validate this in a pairwise manner rather than a regional manner. So what we did is we computed functional connectivity. We got the functional connectivity data from the same subjects, and we reduced that to the granularity of 17 Yale systems, and we did spectral DCM. So that gives us a matrix of effective connectivity. So essentially a asymmetric functional connectivity matrix. And then we downsampled our navigation efficiency matrix to the same resolution of Yale systems. And essentially, we correlated the asymmetry of these two matrices. And we found a really nice agreement. So here in the horizontal axis, we have a measure of asymmetry that is essentially derives exclusively from the embedding of regions in the structural network and the interaction of this embedding with a propagation strategy, navigation, or communication model. And this has a really nice agreement with the asymmetry that we get from DCM, from resting state functional connectivity. So we thought that this was a very nice result. Something else that we did was try to predict human behavior from brain network communication matrices. So we used participants from the Human Connectome project for this paper. And we computed a set of behavioral dimensions. So we computed communication matrices for them, and we computed a set of behavioral dimensions. And we used the communication matrices to predict the behavior using machine learning. So this is a summary of the results. In the horizontal axis, we have different input matrices. So most of these are communication matrices, but we also have functional connectivity here in green and structural connectivity here in pink. And on the vertical axis, we have behavioral prediction accuracy. So what we see is that functional connectivity was by far the best predictor of behavior. And we think that it could be because of different reasons, but we think that the reason here is that functional connectivity is actually capturing the interactions between regions that give rise to behavior, whereas the communication matrices are a model of that. So this gap, to me, tells us that the models are not quite there yet. But the key thing is that communication matrices consistently outperformed the structural connectivity matrix. So in other words, by getting our structural matrix that only had direct monosynaptic connections and simulating communication to get a communication matrix that accounts for polysynaptic signaling, we could really narrow the gap between the predictive utility of FC matrices and SC matrices. Okay, so in summary, I hope I showed you that communication models can help us understand brain function. I hope I convinced you that there are many alternatives to shortest path routing, that should be considered in when trying to understand information transfer in the brain. And this is especially because we still don't know which of these models or future models that are going to be proposed best describes neurosignaling. And we're only going to, I think, make headway in this goal by more studies taking into account more models, so we can form a better picture of which of these models are more predictive of brain function in different contexts. And finally, a very important point is that all of the models that I focused on in my presentation are topological models. So they only take into account the topology of the human connectome. And this is, of course, not the whole story. There are also dynamical, statistical, and information theoretical approaches that can help us understand information transfer in the brain. As I said, this talk is part of the 2021 OHBM Symposium on Communication in Brain Networks. There are 3 other fantastic speakers, and I encourage you to attend all of these talks and also come to the live Q&A sessions that are going to be held in these dates here. Okay, thank you. Bye.
Hello, my name is Bertha. I work at the Network Neuroscience Lab at McGill University. Today, I will talk about some works that study the effect of the heterogeneity of the brain over the structure-function coupling and over the propagations of signals through it. From previous research, we know that the myelin content, the cortical thickness, and many other structural and functional properties of the brain change from unimodal to transmodal regions. So let's start with the question, what is the effect of this heterogeneity over the signals that travel through the brain? To answer this question, we consider the simplest trajectory that a pair of nodes could use to communicate, the shortest path. But first, using diffusion-weighted imaging and deterministic streamlined tractography, we were able to obtain the structural connectivity. And computing the Pearson correlation between both time series, we got the functional connectivity matrix. So from the structural connectivity matrix, we derived the weighted shortest paths, and from the functional connectivity, we got the gradient that ranks the brain along a continuous gradient from unimodal to transmodal regions. Then we annotate the paths according to the functional hierarchies. So each path could be seen as a sequence of hierarchical values, one for each node along the path. The functional hierarchy assigns a real value to each node. But using the cumulative distribution, we bin the nodes into 10 equally sized groups, where 1 will correspond to unimodal regions and 10 to transmodal regions. Let's see an example. Here we see all the different paths that connect regions 9 and 2. Each line corresponds to a path with a different number of steps that occur with a different probability. When we do the same for all the combinations of source-target hierarchies, we found that the shape and the hierarchies visited along the path would depend on the source and target hierarchies. Some paths follow a monotonic trajectory, while others, especially when the source and target hierarchies are similar, will be more likely to follow a U-shape. What causes these weird U-shapes? To explore the nodes that are changing the orientation of the paths along the trajectory, we calculated the average slope of all the paths at the nodal level. Once we have the slopes, we got the average slope of all the paths when they pass through a node. If the average slope is positive, it means that the node was sending information up in the hierarchy. If the average slope is negative, it means that it was sending information to lower hierarchies. Organizing these values by resting state networks, we can see that nodes from the default mode network and limbic network tend to direct signals to lower hierarchies, while nodes from primary sensory regions and attentional networks have a mean slope positive. The slope of a path allows us to identify areas that redirect the trajectories, promoting the detours that we saw as U-shapes on the last slide. For example, If the slope changes from positive to negative, it means that the node where the change happens is connecting two low hierarchies. And we call this point a turning down point. If the change in the slope is from a negative to a positive value, the node is redirecting the information from a high hierarchy to a high hierarchy, and we call it a turning up point. We can see that nodes that belong to the default mode network have the highest probability to be turning down points, while nodes that belong to the dorsal attention have the highest probability of redirecting messages up. So nodes that belong to the default mode network act as mediators between lower hierarchies, while nodes that belong to the dorsal attention bridge the communication between higher hierarchical areas. The next question I want to address is, what is the effect of brain heterogeneity over the structure-function relationship? When we compare the properties of the structural and functional networks, we find that central nodes in the structural network tend to be central in the functional network too. But there is a discrepancy. If we divide the functional weights as the pairs that are structurally connected and the ones that don't have any structural connections, we find that the pairs of nodes that are structurally connected have a greater functional weight. But from this division, we get a lot of pairs that are not structurally connected, but have a functional connection different from zero. What causes the discrepancy between the structure and function? Well, it could be that two nodes that are not directly connected over the structural network through a combination of multiple elementary interactions over the anatomical network get functionally connected. So in this work, we are going to consider that functional interactions could emerge from a combination of different mechanisms over the anatomical network. We constructed a multilinear regression model to predict the functional connectivity from three predictors: the Euclidean distance, the shortest path length, and the communicability. We consider the Euclidean distance as a predictor because it assumes that two nodes could interact just by proximity. We consider the shortest path because it is the most efficient way that two nodes can use to interact. And finally, we use communicability because it assumes that two nodes are going to communicate through a diffusion-like strategy. To support the hypothesis that the structure-function relation could be heterogeneous across the brain, we predicted the functional connectivity node by node, using one row at a time in the regression model. We measure the coefficient of determination between the predicted functional profile and the empirical functional profile, and in this way, we quantify the correspondence between structure and function for each node. We found that the structure-function coupling is not homogeneous across the brain. Some regions show a high correspondence while others a little one. The spatial arrangement of the coupling looks a lot like the gradient that organizes the brain from primary sensory motor regions to transmotor regions. Actually, when we plot the relationship between structure function versus the functional gradient, we found that they are anti-correlated. The structure function correlates very nicely for unimotor regions and decreases as we go up in the hierarchy. Now, we know that functional connectivity patterns fluctuate in time in a non-random way, in a very organized manner that is related even to behavior. The question is, how does a structure-function coupling fluctuate over time? Well, first, we need a functional connectivity that changes on time. So we derived the dynamic age-centric functional connectivity using a recently published method. For this, we get the ball time series for individual brain regions and get the cofluctuation for each pair of nodes, resulting in a time series for each pair of nodes. A different way to see this is each point on the time series will be a point on a cofluctuation matrix. So we can transform all the time series into cofluctuation matrices. We then apply the multilinear model, the one that I presented before, to predict the functional co-fluctuation at each time point from the distance, the shortest path length, and the communicability. The coupling between the predicted and empirical co-fluctuation is quantified with the coefficient of determination, and we will have one value for each node and one column for each time point. When we see the Pearson correlation between the static structure function coupling and the dynamical one, we found that it follows a uniform distribution, showing a variability that goes from positive values, which means that they are very similar, to negative values, with the means around 0. At a nodal level, we will have one value for the static coupling, and for the dynamic coupling, we will have a vector of different values, one for each time step. We can imagine the dynamic coupling as a time series that fluctuates around the static coupling. So we can count how many times the dynamic coupling has a greater value than the static. We get the probability that the dynamic relationship is greater than the static and found that this happens with a greater chance for the intermediate functional hierarchies. A different way to compare the static and dynamic coupling is by making an analogy with darts. If the static coupling is the target and all the dynamic values are the attempts, we can ask how accurate and precise is the dynamic coupling in comparison to the static case. We can compare how similar are the dynamic values to the static and how scattered the dynamic values are around the static value. Using these three definitions, we can compare how the dynamic coupling deviates from the static value and see that the relationship between static and dynamic coupling is not uniform across the brain. It depends on the functional hierarchy, and it shows the greatest differences in the insular regions. We also compute the coefficient of variation of the structure function for each node. These measures allow us to compare different dynamical behaviors. And we found that the most variable fluctuations occur in the middle of the unimodal transmodal hierarchy. There are more things to discuss, but let's recap up to this point. The brain is heterogeneous, and this affects the signals propagating through it. We found that the shape that the signals traveling through the shortest paths will depend on the hierarchy of the source and the target. Some trajectories will change monotonically, while others will show a U-shape. The regions that shape the communication landscape and produce the detours over the U-shaped trajectory are mediators that change the flow of direction of the messages. Regions that belong to the default mode network act as mediators between lower hierarchies, and regions from the attention networks act as mediators between higher hierarchies. We found also that the structure function coupling is strong for unimodal regions and diminishes as we go up in the hierarchy. And when we consider the temporally resolved functional co-fluctuation, the relationship between structure and function fluctuates, exhibiting spatial and topological heterogeneities. Altogether, the relationship between structure and function is heterogeneous spatially and temporally. This is the end. Thank you for listening. All the way up to here, I invite you to visit the poster 2379 during the conference. I would like to finish this talk by recognizing the help and support of the amazing lab I am part of. If you are interested in these works and want to know more, I recommend you to read these papers. Here are the links, or message me. Here is my email. Thank you for your attention.
Thanks for listening to my talk for Virtual OHBM 2021. I will speak today about the link between structural connectivity and functional connectivity from a theoretical perspective. Notably, I will stress how the link existing between structural connectivity on one side, describing anatomical connections between neural circuit components, and functional connectivity on the other side, describing interactions, information exchange between these components, is not a direct one, but is rather an indirect one. Indeed, the dynamics of the neural circuits provide the necessary bridge between structure and function. Structural connectivity shapes dynamics but doesn't fully constrain it, and it is dynamics ultimately implementing functional connectivity. Such a statement may seem self-evident. It is worth nevertheless stressing this role of dynamics as a bridge between structure and function, because dynamics is complex. For dynamic complexity, I refer to the collective nature of the dynamics of neural circuit, which cause dynamics at different scale to be coupled, and critical phenomena such as multistability, phase transitions, or non-local effects of local perturbations to occur. In the rest of the talk, we will follow this path from structural connectivity to functional connectivity and back via dynamics, stressing every time the special effects that derive from the indirect nature of this link. Let's then start from the first of these special effects, which we name here structural degeneracy. Structural degeneracy is linked to the attempt of inferring which structural connectivity could be giving rise to a functional connectivity that we are observing. The problem here with structural degeneracy is that there could be multiple possible structural connectivities that give rise to the same functional connectivity. As an example of structural degeneracy, I consider here a neuronal culture, specifically the attempt to infer the structural connectivity of the culture from the simultaneous observations of the activity of its many different neurons. To the left, you can see time series of culture fluorescence associated to different regions of interest. As a simple inference algorithm, one can think to compute functional connectivity between pairs of neurons, to threshold this connectivity through some suitable threshold, and based on this comparison, to assess whether a structural connection is present or not. The problem, however, is that for an empirical culture, the ground truth structural connectivity is not known. This problem can be fixed if, rather than considering an empirical culture, we consider the simulated neural culture. Here you can see that we were able to generate simulations of the activity of a virtual neural culture and to emulate its culture on fluorescence imaging in a pretty realistic way. You can also see an example of a connectivity inference with true and false positive connections highlighted. We obtain this connectivity using transfer entropy as an information theoretical measure of directed functional coupling. One could certainly devise better algorithms. And indeed, this simple inference problem was also the source of inspiration from a CAD-Goal crowdsourcing challenge we organized a few years ago. However, achieving performance in reconstruction is not really what we care about here today. I want, on the contrary, to explicitly stress structural degeneracy, considering the example of 3 different neuronal cultures with different clustering coefficients at the level of the structural connectivity. From the least to the most clustered culture, all 3 give rise to equivalent bursting dynamics, where you can see synchronous bursts of activity as vertical stripes in the raster plot of the simulated culture. As a consequence of this hypersynchronized burst with equivalent rate, the inferred functional connectivity always display an elevated clustering coefficient. Information about the original clustering level of the underlying structure is therefore lost. In other words, different structural connectivities giving rise to equivalent dynamics give also rise to equivalent functional connectivities. In this example, structural degeneracy is negative since it prevents the structure inference problem. In other contexts, however, structural degeneracy could turn out as a useful mechanism. This is what we are going to see today now, considering another toy model of two brain regions coupled by long-range excitation. This is a work performed in cooperation with Boris Gutkin at École Normale Supérieure and performed mostly by Sophie Benitez-Stulz. So, as you can see, each of the oscillations oscillate, in this case, in phase. However, the phase relation existing between the coupled population can be controlled by various connectivity parameters. By modifying, for instance, the strength of long-range coupling between the populations, one can transform the coupling from in-phase to anti-phase. According to the communication through coherence hypothesis, the relative phase between the oscillations of two coupled populations determines the efficiency of the communication between them. Therefore, as an effect of this simulated cartoon neurodegeneration weakening of the coupling, the efficiency of communication between the two regions will be degraded. However, long-range connectivity is not the only parameter determining the phase relation existing between the populations. By changing not long-range connectivity, but local connectivity, one can indeed obtain equivalent phase relation changes. We consider here the case of the balance between connections from excitatory to inhibitory and connections from inhibitory to excitatory cells within the recurrent circuitry of each of the populations. By changing local connectome strengths, one can rescue once again the phase relation, bringing back from antiphase to out of phase, and finally back again to in phase. An even more paradoxical example is the one of two oscillating regions, now oscillating through the so-called ING mechanism, for which pyramidal interneurons are not necessary, in which I'm modifying not anymore the strength of long-range connections, but rather the latency of these long-range connections, here in the attempt to emulate a virtual cartoon demyelination, for instance. As an effect of the increase of the latency, the phase relation between the two coupled populations will pass again from in-phase to antiphase. However, by strengthening now local inhibition within each of the populations, the phase relation can be brought back to in-phase. In other ways, here, changes of local inhibition strength emulate changes of the latency of long-range connectivity. The reason why this happens are rooted in a mathematical theory known as phase reduction theory and weak coupling theory. In a nutshell, what is changing by changing local connectivity is the phase response curve of the nonlinear oscillating population, which determines the way in which it locks for a given fixed strength of the long-range coupling. More importantly, however, is that while changes of long-range connectivity are difficult to achieve at the global level, one cannot see how the latency or the strength of a long-range connectome could be globally restored. On the contrary, local connectivity can easily be affected by changing levels of neuromodulation. Therefore, our reflection on structural degeneracy brings us to consider a new potential role for neuromodulation, where by modifying pervasively local dynamics, one could obtain a potential compensation of structural degeneration. We now move to a different spatial effect of the indirect link between structural and functional connectivity. We consider here functional multiplicity, which in a sense is the reverse of structural degeneracy, being associated to a situation in which a given structural connectivity does not give rise to just one functional connectivity, but to a multiplicity of possible functional connectivities. The magic trick is here made possible by a non-linear feature of complex network dynamics, namely multistability. We have shown in the past in simple toy models that even simple circuit can give rise to a multitude of possible dynamical modes, each associated to distinct ways to exchange information between coupled populations. This is also the case for large-scale connectome-based models in which not just 2 or 3, but several tens of brain regions are coupled by a structural connectivity matrix inspired from actual tractography data. The local dynamics of each region is described by suitable mean field equations and neural mass models. However, the behavior of a region embedded in a larger network can radically differ from its isolated behavior. In particular, when the global parameters of the model are tuned to be below a critical rating stability but at a safety margin from it, then the model develops a rich multi-stability within different possible patterns of coordinated activation. Each of these patterns give rise to different correlations between regions and therefore to different functional connectivity that will be visited along time, all these without need for plastic or modulatory changes at the level of the underlying structural connectivity, which remains perfectly constant. Let's then prepare the system at a specific state and apply an external perturbation. As an effect of this perturbation, the system may jump to a different state, which will be associated to a completely different functional connectivity network, in such a way that global reconfiguration effects can happen very far away the stimulated state. A striking example of these non-local effects of local inputs can be seen in recent work in which we performed, in cooperation with Daniel Bassett and Leah Papadopoulos, simulations of coupled oscillatory regions on top of a connectome. As an effect of applying a constant DC stimulation to one network node, one observes radical and widespread changes of the oscillatory coherence between coupled regions. Here you can see increases and decreases of coherence even far away the stimulated region I. Interestingly, at the selected working point of dynamics, the entity of the effects of changing coherence can be predicted by functional connectivity to the stimulated site, but not by structural connectivity, indicating that these effects are mediated by collective dynamics, a proxy for which is functional connectivity, but not structural connectivity independent from the chosen dynamic regime. Going back to the landscape of possible functional states, we have already mentioned that under the effect of noise, the system is expected to sample along time different functional connectivity configurations. Because of the structured nature of the system's landscape, we expect reconfiguration of networks to be neither fully ordered, as it will be the case if networks never changed, nor fully disordered, as it will be the case if the network changes were totally random and independent from each other. On the contrary, what we observe performing a recurrence analysis of the simulated resting state of a large-scale model is that the system undergoes an alternation between what we call knots, events of transient stabilization of functional connectivity, and leaps, events of transient fast reconfiguration. These knots and leaps are respectively visible as red square or blue stripes in the recurrency matrices here shown to the left, which gives, for every possible pair of time, the correlation existing between functional networks observed at time t1 and times t2. Knots and leaps exist also in empirical resting state time series, as shown here by this example concerning an healthy aging dataset, where we can see that knots becomes progressively more and more prominent while advancing through age. More sophisticated analysis of the geometry of the random exploration of the space of possible functional connectivities can be performed, notably the trended fluctuation analysis discriminating between walks without structure corresponding to an exponent close to 0.5, and more structured walks with this clustered appearance and persistent memory, characterized by exponent larger than 0.5. Doing a long story short, we observe that the geometry of functional exploration could be actually functional in a cognitive sense, as subjects, independently from their age, displaying more clustered exploration strategies of the space of possible functional connectivities, are also invariantly associated to better cognitive performances. I think you're tired and the time is getting short, so time for a recapitulation. I hope that my talk has convinced you that the gap existing between structural connectivity and functional connectivity must be taken into account when exploring the potential functional impact of functional connectivity reconfigurations. This gap has been materialized into a form of special effects, which are not only curious, but they provide actually novel mechanisms leading to function. Thanks for your attention.
Hi, everyone. First, thank you for taking the time out of your busy schedules to watch my talk. And thank you to Caio, Andrew, and Olaf for organizing this fantastic symposium. I'm pleased to be part of such a prestigious speaker lineup, and I'm excited to share some of the recent work that's been going on in our lab in the last couple of years. So there are many ways to relate functional and structural connectomes, and this is all arising from the observation that the structural connectome, which makes up the white matter pathways in the brain, are what allow the activation patterns to flow through the network and to go from region to region. So that observation means that the structural connectome and the functional connectome, which is based on activation patterns, should be somehow related. And there are many different approaches that have been used in recent years to map structure and function, including linked neural mass models, network control theory, and machine learning, and of course, statistical modeling approaches and graph theoretical approaches, which I'm sure have been discussed in this by the symposium speakers. I'll be discussing a few of these today, but my focus is going to be on their application in populations with neurological disorders. So the effect of pathology on the connectomes is an active area of research, and even since 2009 with these virtual lesion studies where Olaf and his lab looked at how removing structural connections could impact the resulting functional flow of activation through the brain, to recent years where people have been looking at, on a voxel-wise level, how disruptions in structural connectomes could affect functional connectomes. This is a nice little visualization of this idea, where you have two voxels, these yellow voxels connected with a streamline, and they have a high functional correlation or similarity in activation patterns. And then when that streamline or that white matter tract is damaged by a stroke in this case, then the functional profile of their regional time series actually the similarity reduces. And so a lot of activity has been looking at how pathology can impact structural connectomes and thus functional connectomes. There have been many recent studies focused on investigating changes in the structure-function relationship in disease or recovery, including neurological and psychiatric disorders. I've highlighted several here, two of which are based on the virtual brain framework, which is a series of linked neural mass models. And two different studies actually looked at how the parameters that were derived from the virtual brain fitted to patient data were correlated with those patient outcomes. So in stroke, they actually showed that the global coupling parameter that was derived from the virtual brain was correlated with worse motor outcomes. And in Alzheimer's disease, they actually showed that one of their model parameters was correlated with cognitive outcomes in patients with MCI and Alzheimer's disease. So really interesting work in disorders of consciousness has actually showed that the amount of the more severe patients with disorders of consciousness that had more severe outcomes were actually spending more time in functional states that were more similar to structural connectivity than those that were more mildly affected or healthy controls for that matter. And under anesthesia, those differences between the groups went away, and there was actually a lot of time spent in states that were more similar to the structural connectome. So there seems to be some increased alignment of structure and function in lowering consciousness. And in depression, there's also this observation that the more similarity there was between structure and functional connectomes, the worse the depression scores. And then there's also work by Andrew's lab looking at decoupling in schizophrenia. So decoupling between structure and function in schizophrenia. And then a really interesting simulation and real-world patient study showed that damage to the structural connectome, particularly that in the frontal areas, was actually negatively impactful on metastability of the functional activity profiles, and that loss of metastability was related to increasingly worse cognition. So in this area, my lab and I have been using the network diffusion model to link structure and function in different patient populations. So the network diffusion model essentially says that the functional activation flows through the structural connectome like diffusion of a gas through a network. So in the upper left-hand corner, we have here the observed functional connectivity from a particular subject and the observed structural connectivity from that same subject. And then on the bottom left, you have predicted functional connectivity from the network diffusion model applied to the structural connectome. And on the right, you can see the similarity or the correlation between predicted and true functional connectivity for this subject. And you can see that it starts out at a certain level and it increases and then starts to decrease again. And where this peak occurs, the peak between predicted and observed functional connectivity similarity, may tell us something about how closely aligned the structural and functional connectomes are. So the more aligned the structural and functional connectome, you can imagine the shorter model propagation time, which is what we call that. The shorter the model propagation time would be, because you wouldn't need to have very much diffusion happen in the structural connectome to reach the true functional connectome. And the less aligned they are, the longer you might have to let that model propagation time diffuse through the structural connectome to recapitulate functional connectivity. So we applied the network diffusion model to some patient populations, namely severe brain injury and mild TBI, so two different populations. And our hypothesis was that the increased model propagation time would be related to better outcomes after injury. And we actually see this as true in severe brain injury. We compared model propagation time to coma recovery scale, and we actually see that it was significantly positively correlated, which means the longer the model propagation time, the better the person had recovered. And that model propagation time, again, is sort of a proxy for how similar structure and function are. And we compared it to other graph theoretical measures on the structural and functional networks, and none of them were correlated while the model propagation time was. In a follow-up paper, we also saw in mild TBI in a longitudinal framework that those patients who had increases in model propagation time over time in the recovery period also had better recovery of cognition. This is indicating that the increase in model propagation time could be related to better cognitive recovery after injury. We also looked at a similar patient population after mild TBI, and we used the recently developed network control theory approach. And this allows quantification of brain dynamics by identifying commonly recurring states of brain activity. It also allows computing energies that are required to transition between these recurring states. And that's based on the structural connectivity and assuming that the activation flows through the structural connectome like diffusion. So it's kind of a similar framework to the network diffusion model. And we assessed how brain dynamics may change over time in TBI with a set of 49 patients and 39 controls. We see that actually 1 week after injury, there aren't many differences, but at 1 month and beyond, there seems to be a shift in the MTBI patients to prefer the bottom-up visual network dominant states and disfavor the top-down default mode network dominant states. There also seem to be increases in the energy required to transition between states at 1 month that appear to persist somewhat at 1 year follow-up. And the increased transition energies may relate to attention or cognitive impairments, but we'll explore that in future work. To see more details of this, please visit my student Nate Roy's poster. Tracking network reorganization after acute injury or damage can also provide insight as to how the brain recovers lost function. We know from animal studies that adjacent cortex can take over the function of damaged neighbors, but thus far, this type of reorganization has not been shown at the whole brain level in humans. We wanted to investigate if using this novel approach called graph matching could possibly allow us to uncover these mechanisms. So to that end, we took 23 subjects with first-time pontine stroke who had fMRI and motor assessments at 1 week, 2 weeks, 1 month, 3 months, and 6 months after stroke. We applied graph matching to their extracted functional connectomes at subsequent time points to identify regions whose functional profiles may be taken over by other regions at the follow-up time point. So this is a schematic here showing a region that has been remapped to a different region, and this remapping is a measure of the functional reorganization. So for more details on the actual algorithms and the computations, please visit my student Emily's poster and listen to her playlist. So our first hypothesis were that regions with more structural disconnection would have more functional reorganization. And we actually see this as true with looking at subsequent time points from 1 to 2 weeks, 2 weeks to 1 month, 1 month to 3, and 3 to 6 months post-stroke. We actually see those regions that remap functionally more frequently also had more structural disconnection at baseline. So there was this relationship that we hypothesized. We also hypothesized that more functional reorganization would be associated with better recovery. And so we actually see that this is true by looking at a linear mixed effects model with the remaps, number of regions that remap between sessions, predicting change in Fugl-Meyer scores, as well as accounting for scan length, age, and sex. And we do see that the coefficient for the amount of remapping is significant in this model, indicating that those subjects that had more remapping across time also had better improvements in their motor scores. We also see that early remapping, particularly from 1 to 2 weeks after stroke, was related to both baseline motor impairments and eventual recovery over time. So advanced MRI in clinical populations is informative, and we can provide information about how networks change in disease and possible compensatory effects or reorganization that may drive recovery. However, advanced MRI is not always possible in patient populations. Pathology can increase SNR in ways that obscure underlying connectivity changes. Patients can't tolerate long scan times, and advanced MRI costs money and requires high level of expertise. So our next set of investigations was focused on how we could leverage the large number of naturally occurring lesion studies to understand how the brain works generally, and specifically to devise more accurate prognoses or more effective therapeutics. So we want to bring the connectome to the clinic. To that end, several years ago, we developed the network modification or NEMO tool that allows estimation of structural connectome disruption using only clinically acquired lesion masks. We recently updated it to include high-resolution tractography from 420 unrelated HCP subjects. What this tool does is it overlays lesion masks onto a tractography reference set and traces the streamlines that pass through the lesion mask and records the voxels or the regions that the streamline connects. This NEMO 2.0 can provide either voxel level disconnection volumes or regional disconnection values for user-defined atlas. It also runs on our lab's AWS through a browser interface and doesn't require that the user download or learn any software or even use their own computer. And this was all developed by my lab member, Keith Jamison. So there are other tools like the NEMO tool out there, including BCB Toolkit and Lesion Quantification Toolbox, and they've all started to gain some significant traction in recent years in brain behavior studies of the effects of lesions. And so these are just a few of the many studies that are using these types of approaches to estimate structural disconnectivity using only lesion masks, and they include stroke, TBI, and MS. There's also the flip side of this, which is lesion network symptom mapping, which focuses on estimating functional connectivity disruptions from lesion masks. And this essentially takes the lesion mask, averages the bold signal in a bunch of healthy controls over the lesion mask, and then correlates the result with all of the voxels in the brain. And in the end, what you get is a functional disconnectivity volume of the amount of functional connectivity each of the voxels has to the particular lesioned area. And there's, again, been a ton of studies looking at how lesions map to dysfunctions using this method. And interestingly, there's been some science drama looking at how lesion network symptom mapping relates to outcomes after stroke. So if anybody's interested in a little bit of science drama, please take a look at some of these papers. Of course, we couldn't help but throw our hat into the ring, and we adapted an approach developed by Andrew's lab where they were predicting functional connectivity from structural connectivity using a neural network approach. And so what we did was we tagged that onto the end of the NEMO tool output. And so we created an estimated functional connectivity from a lesion mask. And we applied this in patients with MS. So we had 100 subjects with MS that had high and low disability. We estimated their connectivity, and we also calculated their observed functional and structural connectivity. And we actually see that their estimated functional connectivity and estimated structural connectivity using this approach outperform the observed structural and functional connectivity in classifying patients into high and low disability. So please see my postdoc, Jaren Toslu's poster for more information. So, in summary, studying the structure-function alignment in patient populations may better provide us an understanding of what actually causes impairment. It may also shed light on possible compensatory or recovery mechanisms that could then be manipulated to help encourage recovery in these patient populations. So some methods to estimate multimodal connectomes from clinical imaging will allow the connectome and disconnectome to be mapped in the clinic. It may also help us perform more accurate diagnostics or prognostication, and eventually, hopefully, better therapeutic development. So I just want to mention that Frontiers in Neuroscience has this new topic that I'm a guest editor for. So it's open for submission, so please check that out if you're interested. And I'd also like to thank my lab and also mention that we do have quite a bit of posters. So if you're interested in any of the details that I've outlined in this talk, please take a look. Thank you.